Hello, all you cool cats and kittens. It's Nathan Piccoli and Ashley Blumel from Advanced Cell Biology, and today we're here to talk to you guys about phenylketonuria.
What is phenylketonuria? Often abbreviated as PKU, phenylketonuria is caused by the buildup of phenylalanine and phenylpyruvic acid in the body. This is caused by a defect in the gene that codes for phenylalanine hydroxylase, PAH, which is the enzyme that converts phenylalanine into tyrosine. As you can see in the figure, dietary protein is where we get most of our phenylalanine, which is in turn converted into tyrosine and eventually carbon dioxide and water for elimination from the body. In PKU, phenylalanine is not able to be converted into tyrosine, which causes a buildup of phenylalanine and phenylpyruvic acid in the body. As you can guess, an excess of acid in the body is not good for homeostasis. PKU is considered an autosomal recessive genetic condition, and it affects about 1 in 10,000 to 1 in 15,000 infants in the U.S. every year.
Phenylalanine, along with all of the other amino acids, exist in space as D and L enantiomers depending upon the stereochemistry around their chiral center alpha carbon. The concentrations of essential amino acids, such as phenylalanine, are heavily regulated to remain at a steady state level through influx and runout of the amino acid at all times within the body. The phenylalanine levels at any given time can be increased or decreased in many ways. Increased concentrations come from dietary intake of phenylalanine through foods that are mostly high in proteins like meat. Another way the body increases this concentration is through recycling of amino acid stores after the breakdown of polypeptides. Decreases in concentrations come from the integration of amino acids into proteins during translation, oxidation to tyrosine by phenylalanine hydroxylase, or conversion to other metabolites such as phenylacetate. As I just stated, the conversion of phenylalanine to tyrosine is one of the most important regulatory mechanisms to decrease the levels of phenylalanine stores in the body. This conversion is catalyzed by a hydroxylation system consisting of the phenylalanine hydroxylase enzyme, the unconjugated cofactor tetrahydrobiopterin, or BH4, and enzymes that regenerate tetrahydrobiopterin, such as dihydropteridine reductase and 4-alpha carbanolamine dehydratase. Defects in this BH4 regeneration can also affect phenylalanine homeostasis, as this cofactor is an essential component of the hydroxylation system that converts it to tyrosine. Interestingly enough, PKU is only defined as mutations that directly affect the phenylalanine hydroxylase gene rather than any unregulated influx of phenylalanine. There are two different types of hyperphenylalaninemia, or HPA, which include PKU HPA and non-PKU HPA. The defects in BH4 synthesis and regeneration are actually classified as non-PKU HPA. Specifically, phenylalanine hydroxylase catalyzes the stereospecific hydroxylation of L-phenylalanine, which is the main step in this form of phenylalanine degradation. The activity of this enzyme is mostly centered in the liver, but minor activity has been shown in rat kidneys as well. This enzyme exists in humans as a mixture of tetramers and dimers and is comprised of 452 amino acids. The enzyme requires the tetrahydrobiopterin cofactor as well as molecular oxygen for proper functioning within the body. Phenylalanine hydroxylase can be divided into three functional domains. The regulatory domain contains a serine residue, which is most likely involved in activation of the enzyme by phosphorylation. The catalytic domain, with the active site for phenylalanine entrance and hydroxylation, contains a 26 to 27 amino acid motif responsible for cofactor and ferric iron binding. The C-terminal domain is thought to be associated with subunit binding to create the three-dimensional conformation for correct function of the enzyme. It is thought that phenylalanine hydroxylase is subject to control by cyclic AMP-dependent protein kinase phosphorylation and dephosphorylation processes, which would make sense as most of our phenylalanine comes from dietary intake. It has also been shown that manipulation of the BH4 interaction could regulate the activity of PAH as well. The human phenylalanine hydroxylase gene spans roughly 171 kilobases and contains 13 exons within the gene. This gene encodes for a polypeptide of 452 amino acids, which is nearly identical to phenylalanine hydroxylase, except for a few minor post-transcriptional modifications that need to happen. PKU is caused by mutations at this PAH locus. Many of these mutations could be neutral or result in no phenotypic change. They could also be pathogenic if the mutation causes disruption to the enzyme structure, which would affect the enzyme function, as changes in structure lead to changes in function, as with all proteins. The gene has great allelic variation, and pathogenic mutations have been described in all 13 exons within the locus and the flanking region where many transcriptional regulators would bind. Mutations causing the disease can be of various different types, with missense mutation being the most common type, where one base is converted to another, which results in a single different amino acid residue. Nonsense mutations, however, cause a very minimal amount of PKU diagnoses. Now that we have discussed the molecular mechanism associated with PKU, we will transition into the medical aspect of PKU, including symptoms, diagnosis, treatment, and demographics. Phenylketonuria signs and symptoms can range from mild to severe. We have listed some of the most common symptoms. The musty odor is usually associated with the breath, skin, or urine, which is a direct result of too much phenylalanine in the body. The neurological problems can include psychiatric problems, developmental delay, mental retardation, behavioral and social problems, and can even become as severe as seizures. The fair skin and blue eyes result from the fact that phenylalanine cannot transform into melanin, which is the pigment that is responsible for hair and skin tone. The most severe form of PKU is what they call classic PKU, and this form results in more severe symptoms and higher levels of phenylalanine in the body. Some mutations do not cause a complete loss of the enzyme, but rather a decrease in its function, which results in milder symptoms and less risk for brain damage.
Diagnosis of PKU is often done in newborns. Every newborn in the U.S. and its territories, as well as many other countries, require that all newborns be tested for PKU to avoid developmental disabilities. This test is performed at least 24 hours after birth and after some protein has been given to the newborn to ensure accurate results. Blood is taken from the heel or inner elbow and tested for an excess of phenylalanine or other chemicals that could be indicative of a metabolic disorder. If an initial positive result is obtained, then further tests are required, usually using blood or urine tests, and they must be completed quickly so as to ensure that the child's health is maintained. Should a child be diagnosed with PKU, genetic testing can be used to determine the specific mutation that caused the PKU and to help formulate a treatment plan that is specific for that patient. It is very rare in the U.S., but sometimes PKU is not diagnosed until childhood or early adulthood. This can be due to generic mistakes such as laboratory mistakes, sample mistakes, etc. But if a child is found to have developmental delays or intellectual disabilities, they are retested for PKU to ensure that they are, in fact, negative. PKU can also be diagnosed during pregnancy, when the fetus is still in the womb. If an expectant mother wants to know if her child will have PKU after birth, fetal cells can be tested for abnormal phenylalanine content before birth. Fetal cells are either obtained abdominally or vaginally. Testing a fetus for PKU is not common practice, and is most common when a previous child has been born with PKU and the mother is pregnant with another. Because PKU is the result of excess phenylalanine in the blood, the most common treatment for PKU is a low-protein diet with vitamin and mineral supplements. As you can see in the figure, foods high in phenylalanine include foods high in protein, such as meat and dairy, as well as aspartame. Unfortunately, protein is a necessary part of a person's diet, and thus must be supplemented for people with PKU. Because PKU cannot be cured, these dietary changes are lifelong treatments, and they are especially important for women in their childbearing years. If an expecting mother has either untreated or uncontrolled PKU, her fetus is at a higher risk for mental retardation, heart problems, abnormally small head size, and developmental delay. Genetic treatments for PKU are currently being researched.
There is a high level of variability in reported incidence of PKU in different regions around the world. The chart below summarizes many of the different regions to provide a comparison of PKU incidence across the globe. The high incidence in Turkey is thought to be due to the high level of consanguinity, or the fact that much of the population has descended from the same ancestor. The low incidence in Japan is likely due to genetic drift in the founding of the island population. Genetic drift is the relative frequency of different genotypes in a small population with respect to the chance of disappearance of particular genes as individuals do not reproduce. Low incidence in Finland is likely due to the negative founder effect, in which there is a reduced genetic diversity which results when a population descends from a small number of ancestors. It is likely that the difference between this and Turkey is a result of the amount of ancestors that actually carried the PKU mutations into their new colonization.
Because discussing diseases in children is often emotionally taxing, we decided to end our presentation discussing some facts about PKU. PKU was discovered in 1934 and has since been found to be the most common in Caucasians and Asians, while presenting most rarely in those of African descent. The heel prick test that's performed on newborns is used to test for over 70 metabolic disorders, including PKU. Because PKU is almost always diagnosed in newborns, normal development is still a possibility in patients with PKU, and if they adhere to their strict diet, there are almost never complications later in life.
This wraps it up for our discussion about phenylketonuria. So Ashley and I would love to take questions or comments in the comments section of this discussion board. Thank you.